Thanks, organizer, for this wonderful opportunity. We are interested in the it's not sure. Okay. The 4 A's, including aging, Alzheimer's disease, autophagy, and artificial intelligence. Recently, we found a small molecule also starts with A. So next time it will be a 5A topic. And I want to introduce you social media influential Betty. So in general, Betty is talking to herself in front of the mirror, and she was even unable to recognize herself. The reason is because she's having one of the most age-predisposed disease called Alzheimer's disease. And Alzheimer's disease affecting so many people. When we are getting to 90s, it has been estimated 30% of them will get Alzheimer's disease. Pathologically, it has the shrinkage of the brain, especially entorhinal cortex, hippocampus, and accumulation of the garbage, like extracellular A-beta plaques and intracellular tau tangles. And those have been proposed as at least one of the drivers of the disease. However, there have been so many efforts to find a drug for Alzheimer's disease, still no cure. And we are interested in whether we can focus on aging as approach to fund the mechanism of Alzheimer's disease, as well as for drug development. And why aging? For example, if you look at Betty, when she was at her 20s, she had excellent memory. She only started to lose memory when she reached to her 70s and completely lost. And if you look at the brain compared with young, there are a lot of garbage inside. And we propose that when we are young, we have a good garbage recycling system to clean the garbage. And to enable it to function well, we need to have a good engine to let the garbage trunk to work. And this is correlated with mitochondria, as Katia Simon also mentioned. So with those in mind, we propose that why we get Alzheimer's disease when we are older, not when we are young, at least for many of the people. It is when we are getting older, then one out of the engine in the trunk at least contribute significantly to the garbage accumulation. And my group aims to use different approaches to repair the damaged engine, like exercise, fasting, medicine, healthy diets, as well as mental health. And a couple of students in my group, for example, Thomas, he is working on fasting plus NAD to treat Alzheimer's disease. Helin works on exercise plus NAD to treat Alzheimer's disease. And Sasha works on the combination of different drugs. If you're interested, take a look. As Katia has excellently explained what the garbage recycling system, autophagy, is. But I want to draw attention a protein called ULK1, which is the driver initiation of autophagy. And this is a key topic in my group. Today in Dr. Luigi Ferrugi's presentation, he also showed that this protein popped out when the individuals have exercise. So the project one is to share with you that is reduced NAD can reduce mitophagy as a reason to cause garbage accumulation in Alzheimer's disease. And due to the time concern, I only want to give you a summary of this work. We used AD human samples, AD iPSC with Zemacader, including APPPS1 and ApoE4, 2 AD mouse models, and C. elegans. And our data enable us to propose that when we are getting older, genetic and non-genetic factors can cause defective mitophagy, driving accumulation of damaged mitochondria. This pathway, I would like to highlight, may not sufficient to cause Alzheimer's disease. We still need A-beta and tau-related ApoE4 or TREM2 to working together and cause neuronal dysfunction and impaired phagocytotic capacity of microglia, a cell type which can also clean the garbage, as well as increased inflammation as evidenced by this NLRP3 caspase-1 pathway. This may finally causes neurodegeneration and brain atrophy. So how we show impaired mitophagy if it's not a sufficient driver, but a risk factor of Alzheimer's disease? Because when we pharmacologically, like using NAD or uranexin A, or genetically reinstall impaired mitophagy, we saw improved mitochondrial homeostasis, increased neuronal function, and improved garbage clearance by microglia, as well as reduced intracellular tau and A-beta, and finally, memory improvement. So when we got this paper published, Nature Review Drug Discovery had a commentary. In view of the current failure in drug development, even the recent success on anti-A-beta antibodies, but they only had very minor benefits. The reason is because Alzheimer's disease is caused by many things like tau, A-beta, inflammation, as well as energy deprivation. However, if you're targeting on mitophagy induction, you can dramatically improve all those risk factors. And we believe this could be a more efficient way, rather than to targeting on one risk factor. And here I want to give you a few information on how the small molecule NAD+ can induce mitophagy. Before I do that, I think Professor Torin Finkel's group has did a good job, a lot of pioneering work on how NAD can induce autophagy. So NAD, the small molecules, has been shown to be dramatically reduced in animal models, while the data in humans are a little bit not so strong, but they are still evidence. It could be caused by reduced production as well as increased consumption, like through CD38, PARPs, among the others. To understand how NAD is reduced when we are getting older, it's important to understand how it is produced. In mammals, there are 3 pathways for NAD production, including the tryptophan-based kinase pathway, which is less efficient and time-consuming. Press-handled pathway using nicotic acid, which can cause itching and flushing to some of the people. And the most very well studied recent years, it is a salvage pathway. So NAD being digested to nicotinamide converts to nicotinamide mononucleotide, and finally NAD+. Nicotinamide riboside itself also is another independent precursor of NAD. And the current understanding on how NAD contributes to healthy longevity, including its role in DNA repair through the PARPs, as well as by activate as a cofactor for sirtuins involved in metabolism, neuronoplasticity, as well as CD38 among the others. So colleagues in the field have been using NAM, nicotinamide riboside, as well as NMN as a way to boost cellular and tissue NAD. I want to highlight a few of the molecules here, not only NAD+. For example, cyanuric acid is a neuronal protective intermediate, while quinonitic acid is a neuronal toxicant. For example, this year, in collaboration with our clinician, we had a 12-year follow-up on Alzheimer's disease. We show, for example, if myself and Morten both got Alzheimer's disease when we are at 60s, if Morten's blood level of cyanuric acid is higher, then he has a lower chance to deteriorate his memory compared to me. So this is a protective one. And in collaboration with a geriatrician in Norway, we have been showing that for the hip fracture caused delirium, if the individual has higher quinolinic acid, it associated with like a bad recovery and increased mortality. So highlights the importance of basic research on clinical applications. I want to give you a summary on how NAD can induce mitophagy in addition to autophagy, as Professor Torin Finkel reported before. We showed that when we supplement cells and animals with NAD, it increased SIRT1 activity, which increased deacylation of FOXO3, which can transcriptionally upregulate genes involved in autophagy and mitophagy, like LC3 and BNIP3, as well as BNIX. There are many other colleagues who have been showing how NAD can either increase or potentially also inhibit mitophagy, but this one is stronger, so possibly with a final aim, still mitophagy induction. In Norway, we have been very strong in Winter Olympics because it's a country with a lot of snow, and especially for the cross-country skiing. So we have been thinking about if doing exercise plus NAD can even make a better function to treat Alzheimer's disease. And what we have been doing, led by a PhD student, Helin Wang, so we treated the mice with NAD prix NR, and also forced the mice to do 20 minutes treadmill. And our results showing indeed there was synergistic benefits with a paper in preparation. So then I want to share with you the second one is why mitophagy and autophagy is reduced, also as Professor Katja Simon mentioned. And we have been working on this project for 6 years since I moved to University of Oslo. Due to time concern, I want to give you a summary. We used a lot of human samples. For example, we were able to get access to a Norwegian healthy aging cohort. We have around over 100 people, but around 21 to 22 have 4 years before and 4 years after data, serum and CSF. There's age-dependent reduction of ULK1. And also, we were able to get access to 500 people-based, very well-documented Alzheimer's disease clinical cohort. It contains Alzheimer's disease, mild cognitive impairment, and control. And many of them have serum and CSF samples. And this piece of data also shows there is an dramatic reduction of ULK1 when in the AD group. And we also used a big post-mortem brain cohort from King's College London. Brain stage 5 to 6 is very late stage of Alzheimer's disease, and ULK1 levels was also reduced. So our data in the humans show ULK1 is dramatically reduced in aging and Alzheimer's disease. And to understand this is not a bystander, but a driver. So we generate a ULK1 mouse model, and overexpression of ULK1 can reduce A-beta-induced memory loss and tau-induced memory loss. So in conclusion, we explored the mechanism on why autophagy and mitophagy reduced in aging as well as in Alzheimer's disease. Reduced ULK1 could be potentially one of the reasons. Because when we genetically and pharmacologically increase ULK1 activity, we increase autophagy and increase the PINK1-based autophagy as well as FUNDC1-based mitophagy, those two mitophagy pathways. And finally, we saw improved memory in the animal models we have been working on. In my first two projects, I hope I have convinced you the reduction of mitophagy is a risk factor of Alzheimer's disease. Because of this, we have been aiming to developing drug candidates targeting mitophagy induction. And so this project, we're using AI as a method. I think Alec has already convinced you the importance of AI, but I want to give you a summary of this work we have been working on, which published last year. So in general, in 2019, we presented 19 mitophagy inducers. We excluded those can induce mitochondrial damage, like FCCP or rotulin. And then our collaborators has been using vector representations of molecular structures, pharmacofingerprinting, and conformal fingerprinting, and built this algorithm and trained in online database. And then we went access to a University of McCall based small library. If we with one as a best match, we set a score of 0.75, which enables the machine to present top 18 small compounds. And then we asked whether those molecules really induce mitophagy or not. And we went to use this mitokema-based HeLa cell line we got from Torrent Finkel's lab with Dr. Sun Nuo. And then 8 of them were able to induce mitophagy at a dose of 10 micromolar. And then we asked whether those molecules can really induce mitophagy in neurons and improve memory in a single C-Alex model. With a cutoff of 200 micromolar, 2 molecules were able to fulfill the 2 criteria. And then we subject to an AD mouse model. And after 2 months treatment from 12 months, and we saw induction of mitophagy in the brain, a reduction of both tau and A-beta pathologies, as well as increased memory. And we are now working with our colleagues to optimize the structure pending for patent application. Encouraged by this thing, we wondered, are there any small molecules maybe have similar or even better results? This one is supported by a molecule and was led by a postdoc or fellow, Shu Qingcao. She lived in Thailand. So she found that there are a lot of epidemiological regulation studies showing when you eat a lot of colorful fruits, it correlates with better quality of life and a lower chance to get dementia. And she wanted to know whether this is a coincidence or because there are still some molecules can be potential anti-AD drug candidates. And she isolated many compounds, and with the lead one called EFFAA. So she showed that in this mitocQC, mitophagy mouse model, we saw a dose-dependent increase of mitophagy in the brain of the Y-type mice, as well as increased phosphorylation P60, ubiquitin 65 as a marker of PINK1-based mitophagy. We did a lot of experiments, but I want to share with you this 3D human brain in a dish. It's AD iPSC to differentiate two neurons and astrocytes. Then added the red ones will be here, microglia, and culture them together in collaboration with Dr. Hansen Cho. So we showed that this model recapitulates tau pathology as evidenced by high levels of p-tau 217, and our molecule EFFA dramatically reduced tau pathology. It also reduced inflammation as evidenced by lower migration of the red microglia into the center, possibly because of reduced A-beta plaques, A-beta peptide, as well as lower inflammation. We are so excited on those results, so then we used more than half a year and worked with a Dutch company, tried to ask what is the pharmacokinetics of this molecule, whether it can pass the blood-brain barrier. And we are so fortunate to show that after treated in rat, and the half-life is 10.1 hours, so close to that 12 to 24-hour half-life preferred by the pharmaceutical companies. And we also showed that after 2 hours injection through the IV, we were able to detect this EFFA in the cortex region of the mice, indicating it was able to pass the blood-brain barrier. Since in Norway, we are a country with a lot of good sea resources, including Antarctic krill oil. In collaboration with my former head of department, Hilda Nielsen, we showed that the krill oil was able to reduce wrinkles in C. elegans, increase the resilience of dopaminergic neurons after alpha-synuclein exposure, as well as increase lifespan and health span. And I was very fortunate to be involved in translate the basic research to the market. So as a chief scientific officer, we have been developing this NYO3-based krill oil, and the key main ingredients are here. And we actually contribute to 60% of the krill oil market in mainland China. And last year, our sales revenue was $82 million US. And in the past few weeks, because of it reached over 1 million per day because of the radiation wastewater into the sea. So here I want to give you a summary of the work. We showed that the NAD can regulate mitophagy in addition to autophagy, and which contribute to neuronal protection. And this pathway is dampened in premature aging, normal aging, as well as in Alzheimer disease. Targeting on defective mitophagy could be a drug able for Alzheimer's disease. The reason for reduced garbage clearance in Alzheimer's disease as well as in normal aging could be at least partially due to a reduction of the protein ULK1. And we have been using AI as an approach to prepare drug development. And we found small molecule like in passion fruit, which can pass blood-brain barrier and reduce memory loss in Alzheimer's disease. Because we do not normally get Alzheimer's disease, Parkinson's disease, ALS, until we are at a later stage. So my postdoc, Sophia Lothrop, who did her PhD in Aarhus University with Tina, who is sitting here. So she was asking why aging is the primary driver of all those neurodegenerative disorders. And we propose that when we are young, we have the NAD mitophagic access to maintain the quality. However, when we're getting older, NAD reduction, accumulation of damaged mitochondria, which is not sufficient to cause Alzheimer's disease, but we still need the ApoE4, TREM2 combined with this to show the AD clinical features. This also applies to Parkinson's disease, Huntington disease, and ALS. In view of the importance of autophagy in... Yeah. Okay, yeah. So I just want to go down. That is, in Norway, we have this Norwegian anti-AD network, as well as the Norwegian Center on Healthy Aging network. And finally, I want to close my talk to introduce you Ruth. She is 100 years old, and she was pissed off that she was unable to drive because her driver's license was suspended. And she now tried to do the scooter. This shows really we can achieve a healthy quality of life. So finally, thank the organizers, especially many years work together with Morten, Wilhelm Bohr, among the others. So thank you so much for your time. Yeah.
Thanks, Evandro, for a very interesting talk. And